I'm sure, madam.
Yes, sir.
So, good evening, everybody. So, today I welcome to the resource person Lakshmi Murthy ma'am and our academy president Professor Ayappan sir and academy chief executive officer Dr. Ramesh sir and all attendees and all teachers and the lecturers. So, ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವರು ರೆಗ್ಯುಲೇಷನ್ ಆಫ್ ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಸೊ ಕಳೆದ ಒಂದು ವರ್ಷದಿಂದ ಲಕ್ಷ್ಮೀಮೂರ್ತಿ ಮೇಡಮ್ ಅವರು ನಮಗೆ ನಮ್ಮ ಅಕಾಡೆಮಿಯಲ್ಲಿ ಉಪನ್ಯಾಸ ನೀಡುತ್ತಿದ್ದು, ಅವರ ಬಗ್ಗೆ ಹೆಚ್ಚೇನು ಹೇಳದೆ ಸೊ ನೇರವಾಗಿ ಉಪನ್ಯಾಸ ಕೊಡುವುದಕ್ಕೆ ಸನ್ನದ್ಧರಾಗಲಿ ಅಂತ. ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you so much. Warm welcome to all the students and the lecturers who are part of this event today. I wish the members of KSTA, Dr. Ayappan sir and all the other members, the students and the teachers, wonderful 2023. Let it be a year where we increase our knowledge of science and technology, and let us take more interest in the field of science, and let us also develop a scientific temperament. So today, without any delay, we are going to go to a very important topic in the chapter called as Molecular Basis of Inheritance, which is part of the unit of Genetics and Evolution, which is part of the second year pre-university biology, or we can consider it as class 12 CBSE biology. So before we go to this particular topic, let us try and understand what the topic actually means. When we speak about a gene, okay, gene has got multiple ways of defining it, and even to this day, even after almost 50, 60 years after the field of genetics underwent a drastic change, we find that still we are redefining gene. If we take it to the simplest level, we can say gene is going to be a segment of DNA that is flanked by a promoter on one side and a terminator on the other side. That is, to the date, the most accurate description of a gene. Of course, we can go to the functional aspects of gene and say gene is going to be part of DNA that codes for a particular trait. Or we can also say that gene is a part of DNA that carries the recipe for a particular polypeptide. Now, without going into too much of a debate regarding the description or the definition of a gene, let us find out in a simple manner by taking some example what a gene is and what is meant by gene expression. Now, all of you are familiar right from school level with with respect to a hormone that is called as oxytocin, or a hormone that is a sister hormone called as vasopressin. Suppose I consider oxytocin as the hormone in question. Second year students will know that it is also called as the parturition hormone, or it is called as pitocin. Now, this particular hormone which is present, which is oxytocin, oxytocin is the one that is present both in males and females. In males, it is going to be relevant with respect to how it controls certain smooth muscles in the body, including the smooth muscles of the blood vessels. Whereas in females, apart from having the same role during a non-delivery time, during delivery, oxytocin is going to be important for inducing the contractions in the myometrium of the uterus, which is going to help in childbirth. In the same way, oxytocin will also work on the mammary glands, which will help in the release of milk. So we are familiar with oxytocin's role. Now, oxytocin, obviously, it is a polypeptide. It is actually called as a nanopeptide because it's a very, very small polypeptide. It is secreted by the hypothalamus and is released by the posterior pituitary gland, or what we call as neurohypophysis. right? Now, this particular polypeptide definitely is going to have a counterpart in the DNA. That is, there is going to be a segment of DNA which carries the code for oxytocin, and that particular DNA is going to be part of our chromatin network present within the neurons of the hypothalamus. Right now, all of you are also aware that the body cells, whether it is a neuron, whether it is a neutrophil, whether it is a smooth muscle cell, whether it is your bicep cell, all these cells have all been derived by mitosis and differentiation from the zygote. And they have been produced by mitosis, means they are genetically similar to each other. For example, the neuron is going to be genetically similar to your stomach lining cells. It is similar genetically to the neutrophils. It is similar genetically to the cells of your thyroid gland, etc. Or in other words, the gene for oxytocin, which is expressed in the neurons of the hypothalamus, is also a gene which is present in your B lymphocytes, which is present in your helper T cells, which is present in your alveolar cells, etc. Does it mean that this particular gene which is seen in the neutrophils or in your bicep cells, etc., is it going to be expressed in those cells? Do you make oxytocin from the muscle cells? Do you make oxytocin from the WBC? No, you do not. Oxytocin is going to be manufactured by a specific tissue, by a specific group of cells. Similarly, if we can extend the logic, I can put it in a very simple manner to the students. The gene for hemoglobin is present even in your neurons. The gene for insulin is also present in your neurons. The genes for hemoglobin is also present in your bicep cells and also in your WBC, because all these cells are genetically identical. But just because they are genetically identical doesn't mean they produce this protein called as hemoglobin. Or in other words, to even make that example simpler, let us take an example of a restaurant. Okay, ಈಗ ಒಂದು ರೆಸ್ಟೋರೆಂಟ್ ಅಲ್ಲಿ maybe there is a recipe book present. A restaurant ಹೆಸರು, let us say the name of the restaurant is Idli Center ಅಂತ. ಆ ರೆಸ್ಟೋರೆಂಟ್ ಅಲ್ಲಿ ಒಂದು ರೆಸಿಪಿ ಬುಕ್ ಇದೆ. ಆ ರೆಸಿಪಿ ಬುಕ್ ಅಲ್ಲಿ there is a recipe for gobi manchurian also. There is a recipe for pav bhaji also. But ಆ ರೆಸ್ಟೋರೆಂಟ್ ಅಲ್ಲಿ ಆ ಡಿಶಸ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರ್ ಆಗಲ್ಲ, because this is a specialized restaurant called as Idli Center. ಅಲ್ಲಿ idli, sambar, chutney ಇಂಥಾದ್ ಮಾತ್ರ dishes ready ಆಗುತ್ತೆ. In the same way, though your neuron has got the recipe for hemoglobin or the gene for hemoglobin, it will not manufacture hemoglobin. ಹಾಗಾದ್ರೆ ಆ gene missing ಇಲ್ಲ, gene is physically present, but we have decided we will not make the gene for hemoglobin get expressed in your neuron, so we are going to regulate it to see that that gene is switched off. Or in other words, Idli Center restaurant recipe for gobi manchurian ಇದೆಯಲ್ಲ, ಅದನ್ನ ಸ್ಟೇಪಲ್ ಮಾಡಿ ಇಟ್ಬಿಟ್ಟಿದ್ದಾರೆ, ಅದನ್ನ ಓದೋದೇ ಬೇಡ ಅಂತ. In the same way, the gene for hemoglobin is regulated by your neuron to see that it is not expressed. The gene for insulin is regulated by your neuron to see that it is not expressed. Expression and reading, that gene is going to be read, it is going to undergo transcription and translation, and finally the product is made, which is going to be a polypeptide and later folded, etc. Right? We have to make sure that we do not produce that particular polypeptide. And that kind of decision, that I will not manufacture this polypeptide, or I am going to make less amount of a polypeptide, or I will make more amount of polypeptide, etc., is where we regulate or control how the gene is going to be expressed. Right? In your NCERT book, you would have seen there is a very brief mention about gene expression in eukaryotes. In eukaryotes, what you notice is the gene expression, that is, a gene is present and we need to regulate it, that gene expression is going to be regulated at 4 levels in eukaryotes. So regulation of gene expression in eukaryotes is going to occur at 4 different levels. What are the 4 levels in which this gene expression is going to take place, or regulation rather is going to occur? Regulation occurs at the transcription level. What does this mean? This means that, let us say, hemoglobin gene should not get expressed in the neuron, then you don't even do transcription of the hemoglobin gene. Okay, you stop transcription itself. So that when you don't make an HNRNA, naturally hemoglobin is not going to be made. So you stop it at transcription level, do not produce the HNRNA for hemoglobin in the neuron. That is first level. Second level, for example, let us say you have neutrophil, which is a type of WBC. WBC, that is neutrophil, also doesn't want to make hemoglobin, but it is not able to control transcription. And transcription occurs, and the HNRNA for your alpha globin chain, beta globin chain of hemoglobin is made. After that, you don't want it to produce your mRNA. So what you are going to do? Prevent splicing. So you can see that at post transcriptional modification, or what we say as splicing. So at this level you can control it, or at the splicing level you can control it to see that HNRNA is made. We could not control it, but let us see that we do not splice it so that it will not produce mRNA. So maybe your neuron is able to control hemoglobin production at this level. I'm just giving an example, okay? Or your WBC cannot control it at this level, so it goes ahead, does transcription of hemoglobin polypeptides, and finally you will decide not to splice it so that the HNRNA can be destroyed. So you can control it at this level. What other level can you control it in? You can control it at the transport of mRNA. Okay, again to take an example. Let us say I take an example of maybe I can take an example of your biceps cells, skeletal muscle cells. So biceps also does not want to make hemoglobin. So what will it do? It is unable to control at this level. So alpha globin chain HNRNA produce out of it. Beta globin chain HNRNA produce out of it. Right? ಆಮೇಲೆ ಆ HNRNA ಸ್ಪ್ಲೈಸ್ ಆಗ್ಬಿಟ್ಟು ಆಲ್ಫಾ ಗ್ಲೋಬಿನ್ mRNA ರೆಡಿ ಆಗುತ್ತೆ, ಬೀಟಾ ಗ್ಲೋಬಿನ್ mRNA ರೆಡಿ ಆಗುತ್ತೆ. But still I can control it. How do I control it? I will not make sure, I will make sure to see that the mRNA is not transported to the cytoplasm for translation. Okay, so even at this level, I can control the production of hemoglobin and make sure that I don't make hemoglobin. So some cells will see that they are unable to control at this level, but they can definitely control that the recipe does not go ahead. Andre ಏನು recipe ಓದಿ HNRNA ಮಾಡಾಯ್ತು, ಆ recipe ನ modify ಮಾಡಿ mRNA ನು ಮಾಡಾಯ್ತು. Now they can control it and see that that recipe does not go to the shelf. ಅಂದ್ರೆ ಏನು ರೈಬೋಸೋಮ್ಸ್ ಹತ್ರ ಆ ರೆಸಿಪಿ ಕಳಿಸದೇ ಇರೋ ತರಾನು ಕಂಟ್ರೋಲ್ ಮಾಡಬಹುದು. Right? Next, ಅದು ಆಗಬೇಕು ಅನ್ಕೊಳ್ಳಿ. Let us take one more cell of the body where hemoglobin is not manufactured. Let us take your skin cells. Your skin ಕೆಳಗೆ ಹೋದ್ರೆ living cells ಇರುತ್ತೆ. Squamous epithelial cells will be there. So if we go to the skin cells, they also do not have to make hemoglobin, but they are unable to control at this level, or this level. So they have made the HNRNA for hemoglobin, that is alpha and beta globin. They have also spliced it up, and they are seeing that mRNA is made. That also they are unable to control, and that mRNA has escaped the nucleus and gone into the cytoplasm. That also they are not able to control. But they can control it at the final level, which is they can control at translational level. So for eukaryotes, you are going to have a number of levels at which you can control your expression of the gene, or you can regulate it. This is an excellent neat question that can be asked. For example, they can ask you a question. There was a parenchyma cell which was discovered in some plant, let us say it's a maize plant. And in the parenchyma cell, a particular, let us say, HNRNA for manufacture of lignin was discovered, which level did not exist to control the manufacture of lignin in that particular parenchyma cell. So HNRNA ittu already for manufacture of lignin andre, that means this level did not exist in that particular cell. Now it has to control at one of these levels to see that parenchyma cell does not meet lignin. Like this, some questions at NEET level or even CET level can definitely be asked for the students. Right? Now, we do not have to learn any other examples. In NCERT book, they have not given examples what they mean by these 4 levels. I have given you considerable amount of examples for making you understand what we mean by this gene expression control in eukaryotes. Let us now shift to a simpler organism, which of course works in wondrous ways. And that is, we will now move to the world of prokaryotes. And in prokaryotes, we will try to learn about the regulation of gene expression in prokaryotes. Right? Before, by the time I erase the board, why exactly do we need to regulate genes in all the cells of a multicellular prokaryote? Multicellular eukaryote. We need to regulate genes because that itself is going to be but a specialization of cells. When you say that a cell is maturing, when you say that a cell is undergoing specialization, what do we mean? Of course, we mean that it is undergoing structural and functional changes. But if you understand it at the molecular level, you will realize that when we say specialization or maturation of cells, we are basically saying that certain genes are getting getting regulated in those cells, and you are permanently switching off, temporarily switching off certain genes, and that's what makes us have many different types of cells and many different types of tissues. Okay, so please remember this is an important aspect of specialization of cells, right? It is just like saying a cardiologist or a cardiothoracic surgeon definitely knows how to even maybe cook a dish or maybe knows how to repair a car. But if he starts involving into minutest details working of his life, then his specialization as a cardiothoracic surgeon will definitely get hit. So if your neurons have to concentrate on forming synapses for memory, let us say, or in producing neurotransmitters, if they have to concentrate on that, then they have to regulate certain other genes to see that their work does not get affected. So specialization ke regulation ಮಾಡಲೇಬೇಕಾಗುತ್ತೆ gene expression. Right? Let us go ahead now into regulation of gene expression in prokaryotes. Now, in prokaryotes, except maybe some of the cyanobacteria like nostoc and so on, where you may have 2 different types of cells present. Otherwise, what you realize is most of the prokaryotes are unicellular, and even if they are multicellular, you realize that all the cells happen to be the same. So when you do not have specialization of cells, then why should we regulate genes? That is the next question. So in prokaryotes, gene expression regulation is not for specialization of cells. Rather, the regulation of gene expression is going to be mainly to see that resources and ATP is not wasted by the prokaryotes. Okay, to give you an example, let us take one of the most famous prokaryotes which all of us have heard so many times over the 2 years in your PUC, and which is that particular eukaryote, and that eukaryote is going to be Escherichia coli or E. coli. Right? It's a wonderful experimental material that a lot of molecular biologists, geneticists, even people who work with biochemistry, they all love to work with this organism, that is E. coli. Now let us consider E. coli. E. coli is a heterotroph. Okay, so it is not an autotroph, so it has to depend on the environment for its nutrition. Right? Now, E. coli can grow in a lot of different kinds of environments. Right? Lots of hundreds of strains of E. coli present. Let us consider a normal E. coli. So, E. coli works in many different kinds of environments it is able to survive. So, suppose you take E. coli and you give it maybe some egg-based nutrition. Okay, you give it the white of an egg, or maybe you give it a cooked egg, then it is able to survive even on that environment. Suppose you take E. coli and you make it to grow in milk, it is able to grow even in milk. You take E. coli and maybe you put it maybe in potato extract, it can even survive in potato extract. So you find that ಯಾವುದೇ ಥರದ್ದು substrate portrait, nutritive substrate portrait, E. coli, wide variety of nutritive substrates, E. coli is able to survive really well. Now, suppose I put E. coli maybe in potato extract. Then E. coli has to make sure that it is able to derive nutrition from starch. Andre, ಈ environment ಅಲ್ಲಿ E. coli should make an enzyme to digest starch, right? Suppose I grow E. coli in an environment where I give it probably egg white, which is rich in protein. Then in that case, E. coli should manufacture enzymes that can break down the protein of egg white, give it its amino acids for its nutrition. Suppose I grow E. coli, let us say, on milk. Then in that case, E. coli should make the enzymes that can break down the sugar present in milk called as lactose and derive nutrition from it. It should be able to make enzymes to break down the proteins of milk and get amino acids from it and so on. Right? Now, think about E. coli. E. coli ಗೆ ಗೊತ್ತಿಲ್ಲ ಅದು ಯಾವ ತರ ಎನ್ವೈರನ್ಮೆಂಟ್ ಸಿಗುತ್ತೆ, ಯಾವ ತರ ನ್ಯೂಟ್ರಿಷನ್ ಸಿಗುತ್ತೆ, it has no clue. Right? Someday, some of the cells may come in contact with egg white or milk or potato extract or meat and so on and so on. Right? Now, E. coli cannot be making starch breaking enzymes on a daily basis in the anticipation that it may need starch. That means that will be a waste for it. Because manufacture of enzymes are, they are protein based except for ribozymes. They are proteinaceous in nature. And these proteinaceous enzymes, manufacturing them is an anabolic process, right? And we definitely require a lot of ATP input. It's endergonic pathway. So as a result, there's a lot of ATP that is needed to make these enzymes, right? Now what's going to happen is, if let us say E. coli continues to make starch digesting enzymes or protein digesting enzymes or lactose digesting enzymes, and it doesn't even come in contact with certain specific substrates, then it's a wasteful process. All the ATP went waste because we are not going to use those enzymes, which means E. coli should be in a position to understand that yes, I am not getting this amino acid, I need to make this amino acid. I am not getting lactose, so I don't have to make lactase. You understand? So it has to take that decision based on the environment and thereby regulate the genes necessary for making those enzymes. So lactose environment idre lactose digesting enzymes ಮಾಡಬೇಕು. Lactose environment ಇಲ್ಲದೆ ಹೋದ್ರೆ ಆ genes ನ switch off ಮಾಡಬೇಕು. That is exactly what we are going to learn in this particular session today, where we will learn how E. coli can take that decision and it can decide at the molecular level that certain genes can remain switched off because the nutritive substance which can be broken down by those enzymes are not there, and therefore the genes responsible for those enzymes can be switched off. And this decision E. coli can make. And let's learn about one particular such gene regulation in E. coli, which was discovered by two scientists called as Jacob and Monod. I will be showing their pictures to you at the end. We'll do a quick recap using the PowerPoint presentation, and I'll be showing you Francois Jacob and Jack Monod, and they were the people who worked on E. coli, and they realized that depending on the environment this E. coli is going to be put in, it will manufacture the enzymes needed to get nutrition, and it can regulate its genes based on the environment. And they were able to discover this regulatory mechanism in E. coli, and that regulatory mechanism that they discovered is called as the lactoperon. And this lactoperon in E. coli was discovered by the two scientists, Francois Jacob and Jack Monod. These were the two scientists who discovered a regulation of gene expression called lactoperon in E. coli. And let us learn about it today. Okay, we will begin with this now. Right. So, just to give you a recap, E. coli, if it is growing in a medium containing lactose, which we call as milk sugar, if it is growing in a nutritive medium containing lactose, which is milk sugar, it will make the enzymes to break down lactose. If it is growing in an environment where lactose, that is milk sugar, is not available, they will not make the enzymes because the enzymes are not needed, because you don't have lactose to break down. That is exactly what we are going to learn. So, Jacob and Monod were able to do considerable amount of research at the genetic level, a series of experiments that they conducted, and they were able to identify a particular series of segments in DNA on the bacterial chromosome of E. coli. And we will try to look at that. So I hope you all know if you take E. coli, which is going to be present. So when you look at E. coli, there is a nucleoid that is present. That is the bacterial chromosome. I'm sure you are familiar with this. And in that, we are going to take only a certain part of it, and we will be learning it in detail. So a small segment of DNA, I'm going to put it for you on the board. Right? And we will learn about this particular segment, which is called as the lactoperon. Lac here stands for lactose. It is a good neat question also. They can ask you whether lac stands for lactose or for lactogen or for lactase, etc. So lac here stands for lactose, because they are going to make sure that we can actually break down lactose and get our super respiratory substrate, which is going to be glucose, and of course another monosaccharide, which is galactose. So let me give you the gene here. So I'm taking part of that segment of DNA and I'm going to draw it here for you. I am showing it as a double line here. I'm going to show this as a double line, mainly because you know that across the three domains, that is the 5 kingdoms, the genetic material is DNA, and it is always double-stranded. You have very, very rare some of the polyphages which may have single-stranded DNA. So because it is double-stranded DNA, I'm going to show it as 2 lines here for you, right? So what I'm showing here is basically, this is DNA, please remember, this is DNA, okay, which is present here. I'm just showing this as a 2-line structure here. Now, what are the parts that are going to be seen here? Let us understand. So, firstly, and here you have already done molecular biology, and you know that in prokaryotes, between the promoter and the terminator, you have a polycistronic gene that occurs. Polycistronic gene means when you read one gene, you will be able to make more than one polypeptide. Okay, such a gene is called as a polycistronic gene, and each segment which makes a polypeptide is called as a cistron. This you are already aware of. You know the difference between a gene and a cistron. So you are familiar with that. So here we have a polycistronic gene that is going to be present. So what are the parts that are seen here? I just put this briefly to you so that I can fill it up later. Some of you, it may have happened that you may have ignored this part in college. I'll come to that part later. So what are the parts that we have? So here we have a small gene which is going to be called as gene A. We have another small gene present here which is called as gene Y. We have a big gene here present which is called as gene Z or Z. So we have Z, Y, and A that are going to be seen. I will, explain that later. The other parts. Now, A gene, Y gene, and Z gene together, they are actually supposed to be called as cistrons. So A should be called as a cistron, Y is a cistron, and Z is a cistron. ZYA together represents the structural gene of the lactoferrin. Okay, it is the structural gene of the lactoferrin. What does Z produce? So Z is going to be responsible for a particular enzyme. Z is responsible for a particular enzyme called as beta galactosidase. The enzyme that Z is responsible for is an enzyme called as beta galactosidase. Some of the students, to just give you an idea, in a very gross manner, we can say beta galactosidase is going to be very similar to the lactase that your intestinal juice contains. We have a lactase, remember, human beings, you learned that first year. So beta galactosidase is going to be very similar to lactase. That is, it is able to break down an isomer of lactose called allolactose into glucose and galactose. So just remember, beta galactosidase breaks allolactose, an isomer of lactose into glucose and galactose. That is what beta galactosidase does. Y gene, which is present here, Y gene is going to code for an enzyme which is called as permease. Right? Now, what is this permease? Permease will help in increasing the permeability so that a lot of lactose can enter the E. coli cell. E. coli will always have a certain standard level of permeability. There are channels present on the E. coli cell membrane so that lactose can enter from the outside nutritive medium into the cytoplasm. And when permease is present, you will just be increasing the number of these channels so that there's a lot of lactose that can come in. You can just take it as increases permeability towards lactose. Don't say brings about permeability to lactose. It's a very good neat question also, where they can say it brings about permeability towards lactose. No, it does not. It increases the permeability towards lactose. And then we have gene A. Gene A codes for an enzyme called as transacetylase. Now, this transacetylase is going to convert lactose into its isomer called as allolactose. Okay, it just converts lactose into its isomer called as allolactose so that beta galactosidase can break down the allolactose into glucose and galactose. That is what you find. These are the three enzymes coded for by the respective parts here. Please remember there is no line demarcating Z from Y, Y from A. This is only for the sake of depiction on the board that I have put lines there, which also gives you an idea that Z is a pretty lengthy cistron and then Y and A are almost similar in size. That is the cistron size. Okay, let's go ahead. Now what am I going to find? Now I have to come to the promoter. You all know that a gene should have on one side the promoter and on the other side it should be flanked by the terminator. So I am going to have a promoter sitting here, and the promoter is a little bit at a distance from ZYA. This is going to be the promoter gene. You all are familiar that promoter is the site or sequence identified by the RNA polymerase along with its sigma factor so that it can bind to it, and at the end of the promoter it has to start off transcription. You all know that, you are familiar with that. That is the promoter that I have shown here. You understand that? Okay. What else is present between the promoter and the structural gene beginning? There is an additional sequence present, which is not explained in the transcription unit in the in the middle of the chapter. This additional sequence which is present is going to be the switch of the operon. I have to switch on the gene, I have to switch on the gene. No, that switch of the operon which is present is going to be a gene sequence called as O, or standing for operator. So this particular gene sequence is O, or operator, which is between the promoter and the beginning of the structural gene, right? There is a small gene present here which has not been mentioned in your NCERT book. That also I will tell you later, because it's an excellent neat question that can be asked. Even sometimes in JEE, particularly in JEE Advanced, they can ask some of the questions related to gene expression, because you do have genetic material and biomolecules that are explained in the chemistry part. So though biology is not going to be part of the IIT JEE mains or advanced papers, but still you have biomolecules, and definitely this kind of application question can be asked there also about this additional sequence which I would want to speak to you, right? I'll come to those two blank spaces a little bit later. Then I'm going to come to a small little gene present here, and that gene which is present here is going to be called as gene I. Okay, gene I stands for inhibitor gene, or it is also called as the regulator gene. Gene I stands for inhibitor or stands for regulator gene. Now, what is going to happen here? Next to the gene I actually is going to be also a structural gene, but it is part of the regulation site. Okay, it is also a structural gene and it's part of the regulation site. And this is gene I, which is going to be present here, called as the regulator or the inhibitor gene. Because it is a structural gene, that means it is ultimately going to produce a polypeptide, which will then produce a protein after getting folded. Correct? So, because it is a structural gene, it needs to have its own promoter, and that promoter is going to be present here for the sake of simplicity. Okay? 1 second. Just a second. Just a second.
Ma'am, what happened? Hello?
Sorry, there was a brief connection issue. I'm so sorry for that. I'll just continue. I hope you are able to hear me.
Okay, carry on.
Yes, yeah. Sorry for that. So we have this I gene which is present, which is also a structural gene. Don't forget about that. By itself, it is going to be a gene which is going to be necessary to manufacture a particular protein. So that gene is already there. And that gene requires its own promoter. So I'm going to call that promoter for your reference as PI. I'm going to call that as PI. So I have the O gene, which is going to be the operator. This is the operator. I have the P gene, which is going to be the promoter that is present here. I have the I gene, which is the regulator, or some scientists call it as the inhibitor. PI, which is present, is going to be the promoter for the regulator. In your NCERT book, it is just given as P. Please remember what is the difference between this P and this P. This is a promoter only for this structural gene, which is going to be the regulator. Is that clear? Right. What are the two places that I have left blank here? These are the two places which I have left blank. Let us try and understand what those two places are. So here there has to be a terminator for the I gene, which in NCERT book is ignored. And even if you Google some images of gene expression regulation, you will not find this is usually not shown. But there has to be a terminator for the regulator gene. That is ignored sometimes, and the question will definitely be asked in one of the needs, if not this year, maybe some other year. But it is definitely an intelligent question. There has to be a terminator between the regulator gene and the promoter. It has to be present. Next, we find there has to be a terminator on the other side, which is also ignored in your NCERT book. Now, with all these parts that are present, let us go ahead and see how exactly gene regulation expression is going to happen in the presence and absence of the substrate that is going to be lactose. So first I will explain this lactopera in the presence of, or let me go to in the absence of lactose. Let me first explain it in the absence of lactose, and then I'm going to go to in the presence of lactose. So, E. coli, this particular cell, is presently growing in a nutrient medium which does not have lactose in it. Okay, there is no lactose present, which means there is no point in manufacturing these three enzymes, that is beta-galactosidase, permease, and transacidase, right? Because it will be a wasteful process. Transcribing them, translating them, folding them, etc. is going to be such a wasteful process. The enzymes are not needed because the substrate, that is lactose, is not present in the nutritive medium. At that point of time, what is going to happen? At that time, the regulator gene which is present, this regulator gene is going to undergo a process called as transcription. You know, transcription is where we use a DNA template and we are able to create an mRNA. This mRNA is going to be called as repressor mRNA because it is going to make a protein which is called as repressor. So a repressor mRNA is made. This repressor mRNA will now undergo another molecular process called as translation. When it undergoes translation, it is going to produce a particular protein which is called as repressor. I'll repeat again. I gene undergoes transcription and makes an mRNA called repressor mRNA. This repressor mRNA undergoes translation and makes a protein called repressor. Repressor from the front looks like this, but it is a clamp-like protein. Side and it is going to be like a clamp. Okay, that is going to be repressor protein. Repressor protein has the ability to not only read DNA, it can read DNA, that is the sequence of DNA, it can also bind to DNA. So this repressor protein, in the absence of lactose, directly goes goes and gets attached to the operator site because it has got a high affinity to bind to the specific sequence that is present in the operator region. So once it goes and gets bound to the operator region, there is DNA present and now you have put a clamp to it. When you have put a clamp to it, there is going to be a molecule of an enzyme called as RNA polymerase. RNA polymerase will identify the promoter, binds to it, and wants to uncoil the DNA. It has got helicase action, correct? You all have learned it in transcription. RNA polymerase can bind to DNA and also uncoil DNA. It is unable to uncoil DNA because the repressor molecule has got firmly bound to the operator region. So because it cannot uncoil DNA, what happens? As a result, you find that all these three genes, cistrons, for example, okay, they will not undergo any transcription. If they do not undergo any transcription, automatically there is no production of enzymes, which is perfectly what we wanted because there is no substrate for lactose to for these enzymes to work on. You understand that? So in the absence of lactose, Ig produces the repressor molecule which can read the DNA and bind to the operator region. RNA polymerase cannot uncoil the DNA, ZYA do not undergo transcription, and as a result, you find that there is no enzyme production, which is perfect, and you have saved on energy. You might ask the question, aren't we manufacturing repressor molecule? Isn't that a waste of energy? Perfect, that is correct. There is no doubt that you need to spend some ATP to manufacture it, the repressor. But you realize you will be actually saving a lot of energy not manufacturing these three enzymes. So even to conserve some amount of energy, a little bit of ATP being used is absolutely okay for the cell. It is extremely beneficial and sustainable for the cell. Right. Now, the next question that is going to come is, because we find that operator, regulator, its own promoter and terminator are controlling the way ZYA are going to get expressed, we call this set of genes, that is operator gene, regulator gene and its own promoter together are going to be called as the regulatory sequences of ZYA. And ZYA along with their promoter, operator, regulator will be called as an operon. Operon is going to be a set of sequences which will contain the structural genes and the different sequences that can regulate how ZYA can actually work. Another small point to note, which is very, very important for NEET exam as well as JEE Advanced. And what is that? That is, regulator gene does not have its own regulator. You can see next to I, there is no operator sequence and directly promoter is present next to it. What does that mean? I gene doesn't itself require regulation because it is expressed all the time. Whether lactose is present or lactose is absent, I gene is a non-regulated gene. It should work all the time. This question is definitely a good, neat question, right? Now let us go ahead and see what is going to happen to the lactoferron in the presence of lactose. Let us see what will happen to this in the presence of lactose. Now, I was just telling you whether you have lactose or you do not have lactose. This I gene will continue to undergo transcription, will produce an mRNA called repressor mRNA. That repressor mRNA with the help of ribosomes undergoes translation, and a repressor protein molecule is produced by default all the time. Okay, it is we call it as a constitutive gene or an unregulated gene. It is produced all the time. Now, lactoferron in the presence of lactose, let us try and see. Now, suppose this E. coli is presently growing in a medium where lactose is the carbohydrate available. then that means E. coli should definitely break down lactose to get glucose so that it can break down glucose to get ATP. Now, how is it going to do that? Let us try and understand now. This is how it works. So we have transcription of I going on and finally a repressor molecule is produced after translation. Now remember, when lactose is present in the environment, I've already told you there will be a certain permeability for lactose present, so small molecules of lactose will enter. Now, when you look at lactose, let us say I'm going to draw lactose in this manner. Okay, let us say I draw lactose like this. This is lactose which is present. Okay, I'm just taking it as two hexagons just to show that it is a disaccharide. Now, this lactose has high affinity to bind to this repressor molecule. I repeat, E. coli is presently growing in a nutritive medium which has lactose as the sugar of choice, and that lactose has to be broken down to get glucose and galactose. So, lactose is present and small amounts of lactose will enter into the cell because some channels exist, some level of permeability exists. That lactose, when it comes in, this lactose now has got high affinity to bind to the repressor molecule. There is high affinity for lactose to bind to the repressor molecule. Now, what happens as a result is, as soon as lactose binds to the repressor molecule, this is the repressor molecule. This is going to be the lactose which is going to bind to it in this manner. As soon as lactose binds to the repressor molecule, you will immediately see that the repressor molecule that is present, it is going to immediately undergo. It is going to immediately undergo a conformational change. It immediately undergoes a conformational change. Chemistry, you all have learned how important the structure of your chemicals are. So what was present as a clamp like this, the repressor molecule, as soon as lactose comes and binds to it, it will undergo a conformational change. And this particular repressor molecule now cannot bind. So this is a changed repressor molecule. Why did it change? Because lactose bound to it. Therefore, this is an inactive repressor and it can no longer read the DNA nor can it bind to the operator region. So what happens is operator region is now free. Entire operator region is now free. So what happens? RNA polymerase immediately binds to the promoter region, uncoil the DNA, and you will produce an mRNA. So all these will undergo transcription, and when this entire set undergoes transcription, you will be able to manufacture an mRNA, and that mRNA will now undergo translation. That mRNA will now undergo translation. And when it undergoes translation, you will produce three enzymes. One enzyme is called as beta galactosidase. One enzyme from Y is called as permease. One enzyme from A is called as transacetylase. So that permease will increase permeability, more lactose comes in, correct? And when lactose comes in, you can actually start breaking down the lactose by having transacetylase convert lactose to allolactose. Beta galactosidase can work on the allolactose, get glucose and galactose, and cellular respiration can go on perfectly so that you are able to manufacture ATP. That is how it works. So in the presence of lactose, the enzymes are made. As the lactose amount starts coming down, repressor is going to become free and it can again go and switch off the gene. Right? Now, finally, some small little terms that we have to know. When a gene by default is present in the switched off position and gets converted to switched on position, we call this type of regulation of gene expression as inducible gene expression regulation. From switched off to switched on position, we call it as inducible regulation. Here, who is helping to switch on the gene so that the enzymes are made? That inducer is lactose. Lactose is the one that is helping to switch on the gene. Right? Therefore, lactose is called as the inducer here. And such a gene expression is going to be called as inducible regulation. This kind of regulation is also called as negative regulation because lactose is bringing about its own breakdown. As lactose is bringing about its own breakdown, it is also called as negative regulation. Is that clear? Right? So please remember the neat type of questions which can come. They can ask you whether I gene why it doesn't have its own regulators. They can ask you that it's a constitutive gene which is always expressed. They can ask you why don't they have their own promoters and terminators exclusively? That's because it's a polycystic gene. They can also ask you, questions as to who is the inducer in the lactoperon? Who were the scientists who discovered the lactoperon? These are some of the questions that can come. Let us try and see a PowerPoint for this so that can be a quick recap for you. I'll just give you a PowerPoint for this. And any questions you all have till now, you all can kindly ask me. Any questions that you have till now?
Dear students, please ask any questions about this topic.
I'll be very, very glad to explain any doubts, any questions that you present.
ನೇತ್ರಾವತಿ ಮೇಡಮ್ ಅವರ
ಸರ್
ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದ್ರೆ ಕೇಳಿ ಇವತ್ತಿನ ಒಂದು ಟಾಪಿಕ್ ಮೇಲೆ.
Ma'am, I was very glad to understand about gene regulation. It was very good, ma'am. Even I was not able to understand why there were gaps in that diagram. So you made me understand those gaps. So now I got to know it is nothing but the terminator genes which are present and the promoter gene. Thank you very much, ma'am. You told that Lactoperon concept is a negative gene regulation. Is there any positive gene regulation, ma'am? Any example for positive gene regulation, ma'am?
ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮ್ಯಾಮ್.
Yes, sir. Yes, sir.
ನೇತ್ರಾವತಿ ಕ್ವಶನ್ಸ್ ಕೇಳಿದ್ರು ಒಂದೊಂದು ನಿಮ್ಗೆ ಕೇಳಿಸ್ತಾ ಮೇಡಮ್?
ಇಲ್ಲ ಸರ್, ಕೇಳಿಸಿಲ್ಲ.
Ma'am, you told good morning, good afternoon, ma'am.
Good afternoon, amma. Hello.
Ma'am, you told about Lactoperon concept is an example for negative gene regulation. Regulation. Is there any positive gene regulation example for positive gene regulation?
Absolutely, yes. Any gene expression regulation in gravity, for example, lactoperon itself has got positive regulation. ಅದು ನಾವು ಜನರಲಿ at the BSc level ಹೇಳ್ಕೊಡ್ತೀವಿ. ನೀಟ್ ಲೆವೆಲ್ ಅದನ್ನ ಕೇಳಲ್ಲ ಜನರಲಿ. ಮುಂಚೆ we had the AIMS entrance exam which was separate and we used to teach them. I will just explain that briefly to you. So what we find is now imagine a situation where I have low amounts of glucose and I have high amounts of lactose. Why should we always assume that in a natural environment only lactose is present or absent? You can have various situations. For example, high glucose, low lactose. High lactose, low glucose, etc. There are many situations that can be seen in a natural environment. What you generally find is like this. Even I explain ಮಾಡಿದ್ದು ಇದು negative gene regulation via lactose. Positive gene regulation ಹೇಗಾಗುತ್ತೆ ಅಂದ್ರೆ, let us say When glucose is present, glucose can have a pathway where it can increase its own production.
Okay.
When lactose brings about its own breakdown, we call it as negative regulation. When
Yes, ma'am.
glucose can increase its own production, we call it as positive regulation. When glucose is very less in the environment, environmentally, glucose kadme ittu ankondi. There will be a build-up of a particular chemical called as CAMP, cyclic AMP and the anti. So when glucose is low, CAMP levels will go high. CAMP level when they go high, it means that ATP level is going to be less. CAMP is now going to be part of a large protein which is called as CAMP.
Okay.
That CAP can increase the binding of RNA polymerase to the promoter so that transcription becomes faster. Or in other words, glucose level you want to increase glucose level.
Yes, ma'am.
Low glucose level will have high levels of CAMP. CAMP will make large amounts of a particular metabolite protein called as CAP. CAP is then going to increase the binding of RNA polymerase to the promoter so that transcription becomes enhanced. And when transcription becomes enhanced, naturally what's going to happen is you will find that the regulation is going to proceed to make more glucose. So when glucose increases its own production, we are going to call it as positive regulation.
Thank you, ma'am. Thank you, ma'am.
Thank you. Yes, any other question? Any other question? I think that's it, I think. Questions? Any others? Question? I'll just try to put this PowerPoint. I don't know why today it's not opening. Let me just check. And as soon as I have this PowerPoint, we can just put that and we can just take a look at it. I'm just trying to put that.
Please share me. Please share me using WhatsApp.
Yes, we can do that. Then I can.
I sent to group and the chat box also, ma'am.
Yes, that will be fantastic. That will be really good. That will be good. That will be good. Because it will open up.
End of the session, I sent through WhatsApp, ma'am.
Yes, sir. That will be good.
Also our talent search group also.
Yes, definitely. Yes. So, any other questions you all have, you can just let me know. Otherwise, we can wind up the session.
Thank you anyway, madam. Questions ಯಾವುದು ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಇಲ್ಲ. ಸೊ ತದನಂತರದಲ್ಲಿ ಏನಾದರೂ ಬಂದಂತ ಕ್ವೆಶ್ಚನ್ಸ್ ಗಳನ್ನ ನಾನು ನಿಮಗೆ ಮೇಲ್ ಮುಖಾಂತರ ಕಳಿಸ್ಕೊಡ್ತೀನಿ ಮ್ಯಾಮ್.
Sure. Definitely.
ಸೊ ಆಲ್ರೆಡಿ ನೀವು ಕೊಟ್ಟಂತ ಪ್ರಶ್ನೆಗಳನ್ನ ನಾನು ಚಾಟ್ ಬಾಕ್ಸ್ ಮತ್ತೆ ವಾಟ್ಸ್ಆಪ್ ಗ್ರೂಪ್ ಅಲ್ಲಿ ಎರಡರಲ್ಲೂ ಕಳಿಸ್ಕೊಟ್ಟಿದ್ದೀನಿ ಮ್ಯಾಮ್.
Very good, sir. So let the students answer these questions, and then if they send it to me, I will definitely.
Yes, tomorrow morning I send, ma'am.
Sure, sir. Thank you so much, sir. Thank you.
ಸೊ ಎನಿವೆ ಮ್ಯಾಮ್, ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ತಾವು ತಿಳಿಸ್ಕೊಟ್ಟಂತ ವಿಷಯದ ಮೇಲೆ ಹಲವಾರು ಮಾಹಿತಿಗಳನ್ನ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ನೀಟ್ ಮತ್ತೆ JEE ಮತ್ತೆ CET ಎಕ್ಸಾಮ್ಗಳಿಗೆ ಯಾವ ರೀತಿ ಪ್ರಿಪೇರ್ ಆಗ್ಬೋದು ಅನ್ನೋದನ್ನ ಹಲವಾರು ಉದಾಹರಣೆಗಳ ಮುಖಾಂತರ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದೀರಾ. ಸೊ ಥ್ಯಾಂಕ್ ಯು behalf of KSTA and personally, ma'am.
Thank you. And I need to apologize because today, I used a different setup and there was a small glitch in the middle. I'm so sorry about that. I will definitely share the PowerPoint.
No problem.
Students can go through that. Yes.
Okay. So and also ಅದೇ ರೀತಿ ನಮ್ಮ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ, ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ಪ್ರತಿದಿನವೂ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ಕೊಡ್ತಾ ಇರ್ತಕ್ಕಂಥ ನಮ್ಮ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಸೊ ಅದೇ ರೀತಿ ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಾಗಿದ್ದ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಮತ್ತೆ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳಿಗೂ ಹಾಗೂ ವಿವೆಕ್ಸ್ ವೃಂದವರಿಗೂ ಕೂಡ ನನ್ನ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುವಂತ ಸಾಗುತ್ತಿದ್ದೇವೆ. ಥ್ಯಾಂಕ್ ಯು ಮೇಡಮ್ ಒನ್ಸ್ ಅಗೇನ್.
Thank you so much. Thank you everybody.
ಸೊ ನೆಕ್ಸ್ಟ್ 1 or 2 days ಅಲ್ಲಿ ಮೇಡಮ್ ನಾನು ಅದನ್ನ ಹೇಳಿದ್ನಲ್ಲ, ಫಸ್ಟ್ PUC ಸ್ಟೂಡೆಂಟ್ಸ್ ಗಳಿಗೆ ನಾನೊಂದು ಟೈಮ್ ಟೇಬಲ್ ಕಳಿಸ್ಕೊಡ್ತೀನಿ. ಪ್ಲೀಸ್ ಚೆಕ್ ಅಂಡ್ ಸೆಂಡ್ ದ ಡೇಟ್ ಮ್ಯಾಮ್.
Yes, definitely.
After this week,
And thanks, Netravati ma'am. I'm very glad that you have given a good comment. Thank you.
Yes. Okay, thank you, madam. Thank you.
Thank you.